first session.
Okay, so good afternoon, everyone. My respected teachers and delegates present here. I am Dr. Soumodip Ghosh. I will be presenting a topic called antibody-dependent cellular cytotoxicity. It is a mechanism of cell-mediated immune defense which acts as a bridge between the adaptive humoral and innate immune response with the help of three main players, that is the cell expressing target antigens, the antibody, and the immune effector cells expressing the receptors. So we all know the antibodies have main two parts, the antigen binding fragment and the crystal FC portion, and it acts as a bridge. In mediating the ADCC, IgG, IgA, and Ig plays an important role. Among them, IgG is important because for their activity in natural killer cells, and IgG1 and IgG3 are the most important among IgG subclass of antibodies. Now coming to the effector cells, natural killer cells are the most important part. Apart from them, macrophages, neutrophils, and eosinophils have also a role. Now the effector cells should have a receptor. The receptor types are Fc gamma receptor, alpha, and epsilon, each for specific subset of antibodies. The gamma receptor is the most important part because it is needed for the natural killer cell activity, and there are different subtypes. Due to shortage of time, I will not go into the details. In short, these are 1, 2, and 3. Among them, ABC classification have been done. In natural killer cells, all the receptors are activating type of receptors. No inhibitory type of receptor is present. There are some other minor receptors like KIR receptor and NKG2D receptors. So why do the natural killer cells don't kill the normal healthy cells? Because of the role of class 1 MCH molecules that are present in normal healthy cells, the natural killer cells, when recognize those and downregulates their activity, so keeps the healthy cells safe. Also, neutrophils and monocytes also play an important role by the help of the receptors like Fc gamma R1 and Fc gamma R2A. And in case of monocytes, the receptor is EGFR receptor. Eosinophils, with the help of epsilon R1 receptor, parasites are get opsonized by the IgE and then leading to degranulation and lysis of the parasite. So the main mechanism and stage: recognition of the target cell, FBA portion of the antibody binds the export surface antigen, FC receptor cross-links the effector cell, and then the downstream signaling pathways are activated, killing the target cell via the perforin granzyme cell death pathway. This is the most important pathway where the granzyme B enters the target cell and the DNA fragmentation and apoptosis occurs. Also, there is a Fas and Fas lichen pathway which plays a minor role. These are the this is the chart showing the ADCC effector cells and their mechanisms. And this is a schematic diagram which shows the mechanism of the ADCC that I have told till now. So we have to know how to measure the ADCC activity because we have to know the clinical implication also. First, in 1950s, it was measured by a radio leveling target cells like chromium 51. Later, fluorometric techniques came into place, and now newer assays which use the natural cell products instead of the radioisotopes, that is lactate dehydrogenase and other proteases, are used. More recently, a combined technique is used through single cell computer image analysis, and the most widely used cells for quantifying ADCC is the peripheral blood mononuclear cells and purified natural killer cells. Recently, a immortalized natural killer cell have been performed to go away with the intervariable polymorphism. Now, the major clinical areas where ADCC play a role of ADCC in immunotherapy and vaccine development. In 1961, clients demonstrated that monoclonal antibodies like trastuzumab and rituximab using mouse molecules worked by ADCC activity. Later, further mouse models in vitro studies also showed the same results. And examples of monoclonal antibodies like rituximab in non-Hodgkin's, anti-CD33 like daratumumab in multiple myeloma, HER2 positive breast cancer treated with trastuzumab, and others. But apart from formulating monoclonal antibody, we have to potentiate the ADCC activity by defined mechanisms like supplementation of cytokines, manipulation of the effector cells, and modification of the antibody structure. This is done by three methods: changing the Fc domain glycosylation, removing Fc domain fucosylation. This is the important area of interest now, and by engineering, creating heterodimers of different heavy chains of the Fc portion. Of the oligosaccharides, as I have already told, the fucose sugar unit played the largest role. And if we remove the fucose unit, the increase in ADCC activity shows by 50-fold. And in recent markets, most of the antibodies are fucosylated. So if we remove the fucose in these antibodies, the activity of their action can be increased. One example is mogamulizumab. Also, there is another method for potentiating the ADCC activity by PD1 and PDL1 and PDL ligand. In multiple myeloma, which expresses the PD ligand, it seems that the natural killer cell upregulates the PD1 receptor. And if we block this PD1 receptor, the activity of the natural killer cells increases by tenfold. These are the other mechanisms. I will not touch this because shortage of time, but these are the minor mechanisms like monoclonal antibody plus matrix metalloproteinase inhibitor, killer cell immunoglobulin type receptors, and using of immunocytokines. This is a chart showing ADCC enhancing antibodies which are already approved or in the phase 3 clinical trials. Now, in role of vaccination, as Jagaskandar wrote in vaccines title vaccines in 2018, that the antibodies are two types: neutralizing or ADCC mediating. When we vaccinate a individual, neutralizing antibodies prevent it from entering host cell, prevent the virus or the microbes, and ADCC mediating lyses the cells that have been infected. Now, the current area of interest is creating the universal influenza vaccines, where we stimulate the create antibodies against the same portion of the hemagglutinin antigen, and creates an antibody, ADCC mediating antibody, which is not so much strain specific, and it can be used as universal influenza vaccine. Also, the results of HIV vaccine like RB144 vaccine trial done in Thailand and other subsequent areas, it also proved that the role of ADCC mediated antibody immunization is a major role in preventing HIV-1 infection, though this area needs further study in coming days. Thank you.